Hi guys, Mrs. H here. We're going to be looking at active transport today, so let's go ahead and get started on this. Okay, so at the end of this, we're going to be able to identify what active transport is, the difference between primary and active transport through the cell membrane, what can move in and out of the cell as a result of active transport, and then also endocytosis and exocytosis. So these are some of our vocabulary words. These should look familiar from our vocabulary list. So let's go ahead and take a look at and review what's going on with passive transport. So remember, passive transport, molecules are moving from a high concentration to a low concentration. So in this scenario, you'll notice on the left side of the diagram, we have a high concentration of solute, but a low concentration of water. So the water is going to be moving into the left side. Why? Well, the solute can't move. So the only thing that can diffuse through is water. And so that's not necessarily a good thing for a cell. So a cell has to find a way to compensate for that happening, that natural kinetic energy of molecules, the natural tendency for molecules to move from high to low. And that's where active transport comes in. Because we don't want to, for instance, have a whole bunch of salt build up in our cells. Okay, that's bad. We don't want to have a lot of salt. So we need to counteract that high to low concentration. So remember, with passive transport, sometimes we can, what was I going to say? Sorry, I lost my train of thought. So for example, in this situation, I have a solute that I want to keep in the cell. Well, how do I prevent it going from that high concentration to low concentration? Not all things need to leave the cell. Sometimes we want things in the cell. Sometimes we don't, like potassium, want it in the cell. Salt, don't want it in the cell. So active transport works against the natural tendency for molecules to move from high concentration to low concentration. That's just what they do. That's because of their energy. Active transport is the way the cells do it, to keep the things in the cell that we need to keep in the cell and keep things out of the cell that we don't want in the cell. Because of that, we need energy. Okay, energy is required to move those molecules against that high to low concentration. So really what's happening is we're keeping the concentration high on one side and low on the other, depending on the needs of the cell. So if I want to keep salt out of my cell, I want low amounts of salt in my cell. So I need energy. My cell needs energy to do that. That's why it's called active transport. So what is the energy coming in the form of? It's coming in the form of ATP, adenine triphosphate. This is a nucleotide, okay? Just like we saw nucleotides in our DNA and RNA, this is a nucleotide. So this has those three parts, the phosphate, the sugar, and the base. The base is adenine tri, so there's three phosphates. That's the sugar that is going to be used in order for active transport to occur. And this is that diagram of that adenine triphosphate. Let me get my... So energy is harnessed in this bond. So we know bonds have energy, and in this bond between these two, specifically these two adenines, this is where energy is harnessed. This is where it's located. So if I break this bond between these two, and then I have adenosine diphosphate, and then I have a phosphate, this is going to release energy. And so that's where this energy is coming from. We take this adenosine triphosphate, ATP, we break this bond right here, and we get the energy that we need. Okay, so there's three types of active transport. There's molecular pumps, and these use proteins to, just like we saw with passive transport, and we saw those proteins acting as channels and pumps. This, though, will require energy for it to happen, but it's very similar. Then there's endocytosis, and this is when the cell membrane actually acts as like a little baggie, and it wraps itself around the molecules that it needs and brings it in. And in the opposite, exocytosis is you have the little baggie inside the cell, and it kind of melds itself back into the cell membrane and releases whatever was in the baggie. And we'll look at that in a little more detail. That was just a kind of quick synopsis of each one. So let's go ahead and do a comprehension check, see how you're doing so far. How'd you do? Okay, so let's look at those molecular pumps. Okay, so notice right here we have that phospholipid bilayer. Notice right here, here's the phosphates, here's the fatty acids. So the fatty acids are inside, remember, because they are an oil, they don't like water. And so embedded in this is that protein pump. This is a protein. And so what's going to happen is, here I have a solute, could be sugar, could be salt, could be whatever we need. And so I want to keep, if you notice, I have a high concentration over here. I want to keep that high concentration. So this pump will be activated by energy to allow it to open so that the solute can stay in that high to low concentration. We don't want it to go to equilibrium. So the pump is constantly pushing that solute into the cell to keep the solute in a high to low concentration. And because we're using this ATP, it's called active transport. A primary active transport utilizes ATP. And so what happens is the ATP pumps contain a binding site. It'll have one or more binding site where ATP will actually come and lock in or bind, okay, like a bond. A bond is formed to the protein. There's different classes, P, F, V, and then A, B, C superfamilies, and they're based on the ion that they need. Okay, so one example is a sodium-potassium pump. How does it get its name? Because of what it's pumping. So it's pumping sodium and potassium. So it's going to, sodium's bad for our cell. So it's going to maintain sodium in a high concentration outside the cell, and it's going to maintain a high concentration of potassium inside the cell. So what happens here is, again, it's this ATP is going to bind to the site, okay, that ATP binding site. When it does that, it's going to allow for our sodium here to bind to the site and be pushed out into the cell, or out of the cell, sorry. And then the potassium here is going to be pushed into the cell. So we're moving sodium and potassium in and out of the cell utilizing one pump. And so that is going to use ATP binding to the site in order for that to happen. So let's look at this in a little more details. So there's going to actually have three sodiums are going to three sodium ions and one ATP bind to the pump. And when that happens, it's going to actually allow this side to actually open, okay? And so it kind of acts as a gate where when the sodium, so this actually would be, these are going to come in, and see how this is kind of gated so it can't go through. So the ATP is going to actually allow that to open. So how does that happen? The ATP, that adenosine triphosphate, has to break its bond of a phosphate. And when that does that, it becomes ADP. Now, that energy used is going to be used to disengage or open this gate to push through the sodium. So then that leaves two sodiums inside the cell. So then what happens next? Now we're going to have two of those potassiums. Those two potassiums are now going to be allowed to bind to the site, allowing the potassiums to go into the cell. So once those ions have moved in and out of the cell, the phosphates are released, and then the ADP molecule will be released, and actually your cell will use that in cellular respiration to become ATP, which we'll talk about later. And then a new ATP will bind to the site, and this keeps going back and forth. So then a secondary, so that was a primary cellular respiration, or not cellular respiration, that was primary cellular transport. Now this is secondary active transport, sometimes referred to as co-transport. So these actually use what's called a electrochemical gradient to move molecules. So as molecules move in and out, it creates an electrical charge. And so the charges are what allow the molecules to move through the gradient, kind of like a magnet. So imagine a magnet pulling everything. So the ions are creating like a magnet or a charge to move the molecules. So let's go ahead and look at this one in a little detail here. So a sodium ion are actively transported out of the cell, and the potassium are actively being transported into the cell. So our high concentrations, so because sodium and potassium has these, they're ions, they have their charge. So potassium has a positive charge, sodium has a positive charge. And so the concentrations of the sodium are high on the outside of the cell, while the potassium are high on the inside of the cell. So what's going to happen is these are going to be constantly being diffused into the cell due to the charges. So they're being pulled in. So sodium ions are being pulled into, I said that right. So the sodium ions are going to be diffused into the cell due to the potassium ions outside of the cell, and that's where the concentration gradient is. The negative charge, where we're getting the negative charge is in the cell wall, and so that's why they're constantly being moved back and forth, is because of those charges. And so this is a sodium-potassium co-transport. Oops, I'm sorry, I didn't mean to do that. Let me. So the same thing can happen with glucose as well. Glucose will actually work the same way. So then we have semiports and antiports. This is when two molecules are moving in the same directions. So sodium and glucose are called the semiport, or the symports, and then the ones moving in the opposite directions are called the antiports. So I'm not going to really focus a whole lot. I just want you to understand that there's multiple ways that a cell can move and transport molecules in and out. So when we have the pump where the protein is actually pumping them out, sometimes they're moving because of the positive and negative charges, and then sometimes we have both of them working together in a system. And so this is a pretty complicated system all working together. So you've got one team working here and one team working here, but then everyone's working together. So it's kind of an assembly line going on in this cell membrane. So let's go ahead and take a look at a comprehension check and see how you're doing. Okay, so how'd you do? So now let's go ahead and look at the vesicle transport. So this is when we're going to be using that baggie I used before to move things in and out of the cell membrane. And so vesicle transport is just that. So remember, the vesicle is just that little container that holds particles. And so what happens is, depending on whether it's moving into the cell or out of the cell, so which direction, just basically whether the vesicle is going to fuse to the cell membrane to leave something, or if the cell membrane is going to form a vesicle to bring something in. And that's really the difference between these two. Is one endocytosis, endo in, exo out. So it's moving these particles in and out. Now the big thing here is these are going to be bigger particles. So they can't move through that protein. They can't move in and out of the protein because it's just too big. And so let's go ahead and look at these. So endocytosis can actually be broken down into phagocytosis, which is food, pintocytosis, which is liquid, or then receptor mediated endocytosis, which means they need help to do this. So those are the three types, and all of these things are involving moving something that's outside of the cell into the cell. So we're bringing something in. So phagocytosis is like your mouth, nom nom nom, but you don't have a mouth on your cell. So how does your cell do it? Well, really simply put, is your cell membrane in your cell will actually wrap itself around and encase whatever it wants to bring in. So some solid particle, whether it's food or sugar or a macromolecule, it will literally wrap the cell membrane, because remember, it's fluid and it can move. It's going to wrap itself around whatever it needs and bring it into the cell. And when it wraps itself around it, it's going to make a little small membrane around whatever it is, made of the same material that the cell membrane's made out of. So it's just literally engulfing it in. If you ever get sick, your leukocytes, which is your immune system, it'll actually do that to a foreign body. It'll wrap itself in there so you can get rid of that bad stuff in your body. You can also do this to help digest stuff that your cell needs. So it's a really kind of cool thing. Some organisms actually do it as their food. So if I'm a single cell organism, this is how I'm bringing in my food. Where us as humans, that's not how we bring in food, but it is how we destroy those pathogens that are in our body. So any unknown foreign body our immune system will use this to bring it in. So pinocytosis is the same thing, but we're using it for liquid. So instead of it for being a solid, it's going to be the liquid. And again, we're creating that vesicle with the cell membrane to house it in. These are usually smaller and not as big as vesicles that are created. So the phagocytosis, you're going to use like really big baggies, where this would be maybe a snack baggie. It's really the one of the differences between the two. The other thing is what you're putting in it. So phagocytosis, big solid foods, pinocytosis, liquid. So now receptor aided. So receptor aided means I need something to help this occur. So receptor aided means I'm going to have like these little pits, coated pits, and these coated pits are going to have receptor proteins on them. And so what happens is these receptor proteins are going to be grabbing and binding things to them. Once the receptors are full or coated, then the cell membrane will engulf it. An example of this is when you're cholesterol. We take in cholesterol this way. Your lysosomes, when we're digesting something to release it, again, we're using receptor aided. So basically, what's happening is those receptors are binding to specific chemicals, and it's very specific. It can't just be anything. So the receptor is looking for a specific thing in the environment. It will bind to it. Once all the receptors have been coated, have been binded to the particle, it will create that vesicle. Cholesterol, like I said before, is an example of a receptor mediated endocytosis. So instead of just the cell just going and grabbing anything, they're specifically looking for something in the environment. The receptors are grabbing hold of it. Once they've grabbed hold of it, then they'll make that vesicle with the cell membrane. So if we kind of look at these all side by side, this is what the three different kinds of endocytosis would look like. So then exocytosis is basically the reverse order. So instead of taking things into the environment, I have a vesicle that's inside my cell that's surrounded by a membrane, phospholipid membrane, and I need to get rid of it. So it's waste. And what will happen is that vesicle will actually fuse itself to the cell membrane. And as it's fusing itself to the membrane, it's going to push out the waste in the cell. So exocytosis is the exact opposite. And it can be receptor aided as well. So let's go ahead and do a comprehension check. So how'd you do? Remember, if you guys have any questions on this, make sure you're asking me. Hope this helped you understand active transport a little bit better. And remember, with your Cornell notes, we want to make sure that we're getting the key information. You don't have to write every single word for word down. You can use short fragment sentences. So let's start working on those with our Cornell notes. Bye, guys.